So good day everyone, here again we are back at Markajek School, and now we'll be going into genetic toxicity 2 and considering gene mutation. We have talked about genetic toxicity 1, so now we are going to the second part. In the last video we already talked about these changes being caused by chemicals, and we also talked about these changes in a particular DNA sequence or in a particular DNA segment. Now when these changes occur, they could actually be beneficial to the organism, actually beneficial to the individual, or they could be detrimental, or they could be actually lethal to the human being that is that this mutation occurs in, that this change occurs in. In the last video we also considered the direct interaction with the DNA and all that, and then we talked about DNA damage. Well, now in this video we'll be going into the base substitution, base addition and deletion in relation to the nucleotide bases that can actually be substituted, or can actually be bases being added or being deleted from the sequence. Now these changes in the DNA can actually occur in somatic cells or in germ cells, and it can either be addition of a base pair or deletion of a base pair, or actually substitution of a base. And this can cause this substitution of a base can cause transition or transversion. Now it's transition in the sense that when the nucleotide base is a purine and it's being converted to another purine, like from cytosine to uracil or cytosine to thymine. Or it can be a transversion when it's being converted from a pyrimidine to a purine, or a purine to a pyrimidine. For example, adenine base being changed to this in cytosine. Now that is a transversion. Now when there's a chemical change in the nature of a base or the formation of an adduct, probably as a result of chemical oxidative deamination, this can then change the nature of the base. And when it changes the nature of the base during replication, there could be a substitution. There could be a change to actually repair it, or body system trying to repair that difference, or because it has another affinity for another nitrogenous base. Like when guanine is being changed to 8-oxodeoxyguanosine. Instead of it binding to cytosine, it can then bind to adenosine, thereby causing a mutation or a change. So now the larger adducts that are formed in between the double helix can actually cause frameshift mutation. They can distort the DNA structure and cause frameshift mutation. Now base substitution is actually a point mutation because it just affects a point in the DNA sequence, and then as I said before, they can either be a transition or a transversion. RS can cause oxidative damage, which could be a change to the nucleotide base. Now we'll be considering some examples. I'm going into details of them later. Guanosine to 8-oxodeoxyguanosine, which is a transversion. We'll be explaining how that happens. And then 5-methyl cytosine to thymine is a transition. Cysteine to uridine, that is also a transition. Then adenine to hypoxanthine is also a transition. And then we'll be looking at how this actually works, including how these changes occur after replication. Now something to note is that most chemicals cause mutation or actually cause this change in the S phase of the cell cycle. Now there are actually 4 phases. We have the G1, we have the S, then we have G2, and then we have the M phase. So these are the 4 phases. And then the S phase, which is actually the second phase, is where DNA replication, DNA synthesis, and most of DNA repair occurs. So at that point, that's where most chemicals can actually cause their change before it then goes into G2 phase, before it then goes into meiosis and mitosis, and the cell begins to divide. Now the alkylation addition of the ethanol moiety gives rise to an imidazole ring. Now for adenine, it then affects it to be able to pair like cysteine, which means adenine then pairs with guanine. So now this ethanol moiety is talking about when it gets under imidazole rings. So now this adenosine then has 3 rings. Instead of just having 2 rings, it then has 3 rings, and then it causes adenine to pair like cysteine, and then cysteine to become similar to adenine, and this causes some transversions. Now during replication, there could actually be an incorporation of an abnormal base analog during the replication of the DNA. Now we take for example thymine. Thymine has an analog that is called 5-bromouracil. Now instead of adenine pairing with thymine, During replication, 5 bromouracil can then introduce, and then adenine pairs with 5 bromouracil because it's quite similar to thymine, so adenine will be able to bind to it easily. Now, before we actually go into how this transition and transversion occurs, I want to look a little bit at DNA replication, how it actually happens. So now the first three base pairs is GC, CG, TA. Now there are actually two strands, the two parent strands. Now the hydrogen bonds between the parent strands are broken apart, and then one parent strand on the side, another parent strand on the side, and then now the daughter strands begin to form, introducing corresponding bases, corresponding nucleotide bases that actually correspond with the parent strand, and then it continues like that. The new strand begins to form on both parent strands. Now after that, after both strands has actually been formed, further replication can happen sometime later, and then the parent and daughter strand divides. The parent goes on to form another daughter strand that is complementary to it, and the daughter strand forms another strand that is complementary to it. Now the daughter strand that is being formed is an entirely new strand. There's no parent strand there again, and that is what happens on both sides, and the replication continues on, and that is how mutations are immortalized, are made permanent. Now the first example of the base substitution we'll be looking at here is an example of transversion from guanosine to 8-oxodeoxyguanosine. Now the first the sequence we're looking at is GC, AT, and GC. Now we'll be talking about the GC, the first one actually being affected. Now it undergoes oxidative damage, and then 8-oxoguanosine is formed, and then it pairs with cytosine normally. Then replication of the first strand, that is 8-oxoguanosine. It then replicates, and then it binds to adenosine. Now this is because 8-oxoguanosine has affinity for adenosine, so it then pairs with adenosine. Now there's replication of the second strand, that A that just came in to pair with 8-oxoguanosine is replicated, and then a TA is formed. T is brought in to pair with the A. Now if you look at the overall, I used 3, so now it shows as the codon. In the beginning it was GAG before the oxidative damage. That is the first 3 on the first side. The first strand is GAG. Well, now at the end it becomes TAG, and then when it undergoes transcription to form mRNA, it then goes from GAG to UAG, and UAG is a stop codon. So now because of this change, because of this transversion, it then forms a stop codon, and then the translation or the transcription is terminated where it's not supposed to end. To know that it's transversion that has occurred, now is a GC in the beginning and a TA that is formed. Now you just look at this like this: GC, and then you check what is on the other side, what comes first also. Now the first thing that comes first here is G, and what comes on this side is T. Now G is a purine, while T is a pyrimidine, so that is the difference. A purine is being changed to a pyrimidine. Now we check the second one, cytosine, and the second one at this I at the end here is A. So C is a pyrimidine, while A is a purine. So there's a change now from a pyrimidine to a purine. So that is how we just get to know that it's actually a transversion that has occurred there. Now next we want to look at an example of a transition, and then I'll be talking about adenine being converted to haboxanthin. Now this is also done by an oxidizing agent. So now in the first in the codon, the first base pairing we have there is 18. Now A can be converted to H by these oxidizing agents, and then HT is formed. Now this HT undergoes replication. This strand undergoes replication, and then the first strand, we're talking about the first strand, that is H. Now H can bind like guanine, so therefore it can bind to cytosine. So therefore H binds to cytosine during the replication. Now after this replication and HC is formed, the second strand is replicated, that is C is replicated, and now C is normal, there's nothing wrong with C, so you can then bind with G. Now this H that was formed, the HC that was formed there, if it undergoes replication again, H can bind to either T or can bind to C. When it binds to C, that's when the mutation occurs, and this can go on and on and on, and more of these mutants or more of these changes actually being formed. So here we see a change from AT to GC, and then the codon changes from AUG to GUG. Now how to know this is also a transition? We look at it. Now the first on the strand here is A, and then the last there is G. So now we see it's being changed from a purine to a purine. That is a transition. Now the next that appears with that A is T, and then next that appears there is cytosine. That appears with guanine is cytosine. So that is from a pyrimidine to also a pyrimidine. So that is transition, and that is how we know that change, that base substitution that happened there is a transition. So yeah, that is that about transition and transversion. Now we come to frameshift mutation. Now frameshift mutation is caused as a result of insertion or deletion. Insertion or deletion of a base pair. Now when there's a base pair and there's an insertion or deletion, it changes the codon arrangement, and then when one is removed, the codon is changed, and then it shifts to like cover up for the damage or for the one that has been removed. But now when a deletion happens and then an insertion happens not too far from where the deletion occurs, in close proximity to it, it doesn't exactly change or sends the codon out of shape entirely, but just causes a change where the protein that is being formed now, where the protein that is being formed is just of partial function, but that regulates it or that helps it now so that the codon is not just out of arrangement. Now this insertion or deletion should just be an insertion of just one or two, or deletion of just one or two nucleotide bases, or not of three, because that will take away the entire codon or impute a new codon. Now looking at the example here, we see the normal one first, and then we see the insertion. The insertion is just T, and then the codon shifts to be arranged, and then T comes out, and that T at the end comes out. Now we see an insertion mutation that is non-frame shift. Now there's insertion of 3 complete codons, so that means that 3 complete codon that is inserted now can then code for another protein on its own, and then this causes a difference in the primary structure of the protein, because primary structure of the protein is like the amino acid sequence, how the amino acids are arranged. But now because of this insertion of 3 codons, it then forms a new protein, and then changes the function of the protein, because now the protein that is being formed is different from the original protein that was supposed to be formed there, because of this insertion of a new codon. So that will be all for this video, and don't forget to like and subscribe, and you can use the comment section. Thank you.